plasma membranes are positioned at the boundary of each cell, acting as selective barriers that allow passage of necessary materials into the cell. One of the most important components of the plasma membrane is the phospholipid bilayer. Phospholipids are made up of a phosphate group head and two hydrocarbon tails. The phosphate group, part of the head of the lipid, is polar. This makes the head hydrophilic. The hydrocarbon tail is nonpolar, making it hydrophobic. When phospholipids are exposed to an aqueous environment, they organize into a phospholipid bilayer. The hydrophilic heads surround the outside and face towards the water, while the hydrophobic tails group together in between the layers of phosphate heads. Proteins incorporated into the membrane are called embedded proteins. These proteins go through the membrane and create a tunnel which certain substances can pass through. Membrane proteins can act as tunnels, but they also act as transport, carry out enzymatic activity, signal transduction, cell recognition, intercellular joining, and attachment to the cytoskeleton. The bilayer is held together by hydrophobic interactions, which are extremely weak. This allows the membrane to shift and the proteins to bob up and down, allowing it to make a fluid mosaic. Areas of the membrane with unsaturated fats are more fluid because there is more space between the tails in the layer. Embedded proteins, steroids, glycoproteins, and glycolipids all act to manipulate the fluidity of the protein. Steroids restrict the fluidity of the membrane.
Because the membrane acts as a selective barrier, it is permeable to some extent. The hydrophobic and hydrophilic regions of the membrane affect what is allowed through. Nonpolar, small molecules are extremely permeable because they have favorable interactions with the nonpolar tails of the phospholipids in the phospholipid bilayer. Molecules that cannot pass freely are large or polar molecules such as carbohydrates and ions because of their unfavorable interactions with the nonpolar tails. Molecules that can't pass freely are able to cross through transport proteins. Some act as tunnels while others shuttle molecules across the bilayer. For example, water, which is polar, is able to cross the membrane with the help of an embedded protein, aquaporin. Cell walls act to protect the cells, maintain their shape, and prevent the cell from bursting due to excessive water intake, meaning they maintain the correct turgor. Cell walls are thick and made of a polysaccharide cellulose matrix and lots of proteins. The matrix keeps it reinforced and structurally sound.
There are many ways for molecules to pass through the bilayer, one of which is passive transport. Diffusion is a type of passive transport where molecules go through the layer from an area of high concentration to one of lesser concentration until equilibrium is reached. The cell does not expend energy for passive diffusion to occur. Osmosis is the diffusion of water across a membrane to an area of a lower free water concentration. Facilitated diffusion is the diffusion of small polar molecules and ions with the help of transport proteins down their gradients, which requires no energy. Active transport, on the other hand, requires energy in the form of ATP. Active transport is the transport of molecules facilitated by a carrier protein against the concentration gradient, like the sodium-potassium pump. In order to move sodium and potassium ions across the membrane, an enzyme called sodium-potassium ATPase and the hydrolysis of ATP for energy are used. The pump is able to change shape in order to move either sodium or potassium ions as needed. Large molecules such as sucrose are moved across the membrane by way of co-transport. In this type of active transport, a protein pairs a solute going uphill with a different substance going downhill, allowing for big molecules to pass through. This type of transport is also powered by ATP.